Ladies and gentlemen, good afternoon and welcome to the IAS Lee Gong Chen Distinguished Professor Public Lecture. We are happy to welcome you today at the Singapore Hokkien Hui Kwan Auditorium. I am Stella, an undergraduate from the NTU School of Humanities, and I'm honored to be the MC for today's public lecture. This event is organized by the Institute of Advanced Studies at Nanyang Technological University with support from the Lee Foundation in partnership with the Global Young Scientists Summit Singapore and National Research Foundation. This occasion also reflects NTU's strong and ongoing commitment to advancing women in science, engineering, and research. Through initiatives such as Women at NTU and the promotion of women in engineering, research, and science, the university continues to champion inclusive excellence and support the growth of women across STEM disciplines. Now, before we begin, let's have a group photo together with everyone in the auditorium. To begin with, may I now invite on stage Professor Sam Ziqian, Director of the Institute of Advanced Studies, to deliver his welcome address. Professor Sam, please.
Good afternoon. It's very heartening to see a big turnout for your talk, David. And yeah. Okay, Professor David Baker, 2024 Nobel Prize in Chemistry laureate, professor at the University of Washington, Professor Hanalei Rohola Baker, co-director, Institute for Stem Cell and Regenerative Medicine, professor at the University of Washington. Our guest of honor, Professor Joseph Sung, Senior Vice President, Health and Life Sciences, Dean, Lee Gong Chen School of Medicine and NTU Distinguished University Professor. Distinguished guests, colleagues, students, ladies and gentlemen, good afternoon and a very warm welcome to the Institute of Advanced Studies, IAS Lee Gong Chen Distinguished Professor Public Lecture featuring Professor David Baker and Professor Hanalei Rohola Baker. This lecture is a partner event of the Global Young Scientists Summit, and we are delighted to host you here at the Nanyang Technological University. This occasion also aligns meaningfully with NTU's ongoing commitment to advancing women in science, engineering, and research, championed through initiatives such as Women at NTU and the promotion of women in engineering, research, and science, POWERS in short. To all guests joining us from beyond NTU, a special welcome to our campus, and we hope you enjoy the opportunity to experience our beautiful and vibrant campus. Now, it's my privilege to open today's event, where we gather to explore two extraordinary frontiers of modern science: artificially designed proteins and pioneering advances in regenerative medicine, guided by the expertise and invaluable insights of our esteemed speakers. Let me begin by first expressing our profound gratitude to our sponsor, the Lee Foundation, for their steadfast and generous support. Today's lecture is made possible through the collaboration between NTU's Institute of Advanced Studies and the Global Young Scientists Summit, organized by the National Research Foundation, Singapore. This collaboration exemplifies a shared commitment to nurturing talent, fostering intellectual exchange, and strengthening Singapore's global presence in science and innovation. Since its inception in 2013, the Global Young Scientists Summit has grown into a premier international platform that connects young researchers with leading scientific minds. Now in its 14th edition, The summit continues to champion transformative ideas in science and technology, ideas that tackle fundamental research challenges, enhance innovation, and inspire new perspectives for a rapidly evolving world. At the Institute of Advanced Studies, we see our role as bringing the world's most impactful science to the heart of NTU, and equally, bringing NTU's strengths to the global scientific community. IAS is dedicated to cultivating an environment where interdisciplinary thinking flourishes, where mathematics meets biology, physics engages with engineering, and data science informs chemistry, sparking new collaborations across traditional boundaries. By convening pioneers who have reshaped their fields, we hope to inspire early career researchers to challenge conventions, pursue ambitious questions, and drive discoveries that benefit the society. Today, we are privileged to hear from two scientists whose work embodies the transformative power of such cross-disciplinary thinking. Professor David Baker, Nobel laureate in Chemistry 2024, is a leader in protein design whose work uses deep learning to create novel macromolecular structures with applications in therapeutics and sustainable materials. His research has revolutionized our ability to engineer biological functions once thought impossible. Professor Hanalei Ruhola Baker is a professor of biochemistry and co-director of the Institute for Stem Cell and Regenerative Medicine at the University of Washington. She's a global leader in stem cell biology and regenerative medicine, and her research integrates AI-designed proteins and advanced genomic technologies to pioneer strategies for tissue repair and regeneration. We are especially pleased to recognize Professor Ruhola Baker's outstanding contributions to regenerative medicine, which exemplify the impact and leadership of women in STEM. We are also honored to welcome our guest of honor, Professor Joseph Song. Professor Song is a globally esteemed clinician scientist and academic leader, where he guides innovation across health, medicine, and life sciences. He is internationally recognized for his pioneering contributions to gastroenterology and digestive disease, and for his leadership in integrating clinical excellence with research and education. To our audience, especially students and early career researchers, I hope today's lectures serve as more than an opportunity to learn. Let them kindle your curiosity and strengthen your determination to explore the unknown. The breakthroughs we celebrate today were born from bold ideas, curiosity, persistence, and a willingness to cross disciplinary boundaries. I encourage you to embrace that same spirit as you shape your own scientific journey. Thank you once again for joining us. I wish you an enriching and thought-provoking afternoon ahead. Thank you.
Thank you, Professor Sam. May I now invite our guest of honor, Professor Joseph Song, Distinguished University Professor, Senior Vice President, Health and Life Sciences, and Dean of the Lee Gong Chien School of Medicine, on stage to give his opening remarks. Professor Song, please.
Wow, my goodness, I haven't seen so many people in my whole life before. This is really a very rare occasion that the whole room is packed with people. You see all the people are standing. And there is a tradition in Singapore that before people come into the hall, before the concert or the talk starts, they will tell you if there is a fire, where should you go? So today, I don't know where we should go because all these people are packing up at the back, but I hope there will be a door for our esteemed speakers over here. So once again, a very warm welcome to our esteemed speakers. And I want to start by telling you this is a very special place of the university because the university was built by a group of Hokkien people. That's why it's called Hokkien Hui Guan Auditorium. So they actually Not many of them are very wealthy people. Everybody chip in $10, $1, even taxi driver give a whole day of their income to build this place. And this is the very place where the whole Nanyang University was started about 50 years ago. So we are in a very historical site. When you finish, you may want to take a look at the heritage building outside, which is now a museum. Once again, a very warm welcome, ladies and gentlemen, to this distinguished lecture. On behalf of NTU Singapore, I really have great pleasure to welcome you to this very special lecture. And from the number of people here, Professor Sum told me that he actually did not expect to have so many. It must be your topic and your esteemed honor that attract all these people. So my job is really to tell you a little bit about the two important lectures. Professor David Baker, who is Nobel laureate in chemistry 2024, is pioneer figure in protein design and synthetic biology. As director of the Baker Lab at University of Washington in Seattle and the Institute for Protein Design, he has led a fundamental shift in how we understand and now create proteins. So not just God create proteins, now human beings with the help of AI also do that, and the molecule will mediate almost every process of life. Rather than modifying protein that mediates everything and shaped by evolution, his work really focused on designing entirely new proteins from the first principle. Using advanced computational and deep learning approaches, his team has opened unprecedented possibility in therapeutics, biotechnology, and sustainable materials. So the application is really very wide. His scientific impact is reflected not only in the 600 publications, but also produced over 100 patents and found more than 20 companies translating their discovery into real-world application. Today, Professor Baker's lecture entitled Design of New Protein Functions Using Deep Learning, he will describe to us the recent advances in this rapidly evolving field. He will show how machine learning can be used to design amino acid sequences that fold into desired structure and perform specific functions, protein that evolution has not even produced. But we are actually gathering here a group of biologists and engineers together to listen to this exciting advancement. I remember Bill Gates said with artificial intelligence, all professions will disappear except three. One is not doctor, it's not even teachers, but it's a biologist, energy expert, and the third one I forgot. So today, and the third one must be AI expert. So today, I think we are gathering all the survivors of AI technology here. Our second speaker, Professor Helene Rohola Baker, is professor of biochemistry and co-director of the Institute of Stem Cell and Regenerative Medicine at the University of Washington, Seattle. She is a global leader of stem cell biology and regenerative medicine, a very exciting field in clinical medicine. And she is known for integrating artificial intelligence, protein design, and human stem cells to develop new strategies for tissue repair and regeneration. So when we get old, our organs is failing, she's giving us a spare tire and how to replace the non-functioning parts. Her work also led to breakthroughs in vascular stability, organ regeneration, and cellular reprogramming, with very far-reaching impacts for aging, degenerative disease, and organ failure. You may not know that in Singapore, every 5 years, the government will launch a whole program of research support. We call it RIE. And in the next 5 years, which is under RIE 2030, healthy aging is the grand challenge, and the government is going to put in quite a sizable sum of money into this area. So after listening to your talk, everybody will have a good idea how to apply this grant. So today in her lecture, she will describe AI-driven foundational regenerative medicine. Professor Rohola Baker will discuss with us how the convergence of machine learning, protein design, and stem cell biology is going to transform regenerative medicine from a reactive discipline into a predictive and programmable one. She will present a series of success stories that illustrate how designed protein can act as biological instructions, guiding cell fate, tissue repair, and healing their root causes. Well, I won't waste you more time and delay the lecture. So please join me to welcome Professor David Baker to give the first lecture.
Thank you, Professor Song. Can you please remain on stage? Sorry, Professor May, can you please remain on stage? Sorry. Now we will present the mementos to our distinguished speakers, and may I now invite Professor David Baker to receive the memento. Thank you, Professor Baker. May we now invite Professor Rohola Baker on stage to receive the memento. May I request the professors to remain on stage, please? May I request the professors to remain on stage, please? May I now also invite up Associate Professor Sirin Lim and Associate Professor Xia Kalin to join Professor Sam Tzu Chien, Professor David Baker, and Professor Rohola Baker for group photo taking. You may take your seats now. Now, let's get started with the public lecture. Before we begin the lecture, please take note that the Q&A session will take place after the talk. There are mic runners available for those in the audience with questions. Kindly raise your hand and they will bring the microphone to you. Our first speaker today is Professor David Baker, the recipient of the 2024 Nobel Prize in Chemistry for Computational Protein Design. The title of his lecture today is Design of New Protein Functions Using Deep Learning. Without further ado, let us welcome Professor Baker on stage with a big round of applause.
Okay, perfect. Yeah, I think it works. It works, yeah. Well, you know, there's sort of an asymmetry to the room right now. Is there people sitting on the steps over there, but not on this side? So you're going to have to listen to us for a long time. So I would take advantage of the steps if I were you standing in the back. So let's see. Well, thanks so much for inviting us here, and it's and the really nice introductions, and this is really a great experience for both of us. We enjoyed talking to the students earlier, and we are looking forward to further discussions. And yeah, thanks so much Siren for suggesting that we visit, and to NTU for hosting us. And it's really very special. This is our first time in Singapore, and we have really enjoyed it. So today I'm going to tell you about the design of new proteins using deep learning. And as you know, proteins carry out almost all the important functions in our bodies. And up until recently, the only proteins we knew were the ones that came through natural evolution. These proteins are remarkable. They carry out, like I said, they do essentially everything that's needed in biology. But they're also limited in the sense that they solve the problems that were relevant during evolution, but they don't solve problems that that exist in the world today. Like so, Siren's and I are collaborating on trying to break down plastics and other polymers. Those were just not problems that were relevant during evolution, and so. So I'm going to tell you about today is how we can use deep learning methods now to design brand new proteins. So in a way, the problem of biology is how do we go from the sequences of proteins that are encoded in the genes of our genomes. We know the genetic code that these encode amino acid sequences, and with AI methods we can now predict structures. And sometimes from those structures, we can say something about biological function. So this is sort of the flow of information in biology from the genome to proteins to function and then to interactions. And that's how biology is built up. So what I'm going to tell you about today, though, is not the problem of going in this direction. This is the protein structure prediction problem. But I'm going to tell you about the inverse problem. And here, we start with a brand new function that doesn't exist, like breaking down plastic. We work backwards using the protein design methods I'll tell you about today to designing amino acid sequences that fold up to proteins which have these new functions. And then in the last step, we have a new amino acid sequence. It's brand new. It doesn't exist in the world currently. So there's no existing gene which encodes this protein. Instead, we make a synthetic gene that encodes the protein, and then we put that synthetic gene into bacteria, and the bacteria make the protein, and we essentially extract the protein from the bacteria, and then see whether it does what we designed it to do. And so I'm going to give my talk today is basically a series of examples of the different sorts of problems that we can solve using this approach. We always start with a new function that we want to make a protein to do, and we end up with a protein in a test tube or a protein displayed on the surface of yeast that we experimentally test to see whether the protein we designed actually does what we designed it to do. Now, in a way, my talk is a little bit misleading. In fact, very misleading. It may be the most misleading talk you've ever heard because I'm going to show you these many examples of beautiful proteins that have these beautiful functions, but not all proteins we design actually do exactly what we want them to do. But I'm not telling you about them today, so just keep that in mind. So I'm going to describe a little bit about how the AI methods that we've developed work, and we've borrowed them. These are generative AI diffusion models from computer vision, and the way these methods work, and this is basically image generation methods like DALL-E. And as many of you know, the way these methods work is you start with a large data set, in this case of images, and you noise these images to different extents, and then you train a network, a large neural network, to remove the noise. So like at this point here, to make the best guess about what the actual image was. And then once you've done this, these networks have the remarkable property that you can start with completely random noise, progressively denoise it, and you're left with something that looks like it came from your training set, in this case, a new image. And if you've done it right, it shouldn't be any obvious way to distinguish the new thing you've made from the training data, but it should be completely new. It shouldn't be exactly like any of the images in your set. So we did exactly this with proteins. We took all the protein structures in the protein structure data bank. We noised them to different extents. We trained a model, RF diffusion, to remove the noise. And then once we had done that, we found that we could start with a completely random arrangement of amino acids, progressively denoise this to generate a new protein structure. And these new protein structures looked like they could be protein structures in the protein data bank, but they were actually new. And I should just say that this was the original version of RF diffusion, where the diffusion's happening at the level of amino acid residues. The most recent version called RFD3, and all of our software is available open source, so we released RFD3 a few weeks ago, carries out the diffusion at the level of atoms instead of residues. And that's very advantageous for enzyme design, which I'll be talking about later. So this ability to generate new protein structures by starting from completely random distributions of amino acids and then progressively denoising them is sort of interesting if you care about protein structures, but most people care about protein functions. So we need a way to condition this process on a particular function. So analogous to in image generation, you can say generate an image of a cat sitting on a table. In the case of protein design, you might say design a protein which binds a particular target. So this illustrates that here. So here we have the insulin receptor, and we carry out this diffusion process starting with a completely random distribution of residues, which is here, and it sort of denoises itself into an arrangement of residues in a protein structure which looks like it should bind the insulin receptor. And then, as I said, we make a synthetic gene encoding this protein. We make it in the lab, and we found, and like I said, not all designs bound. We found some designs that bound the insulin receptor pretty tightly, and we've been able to turn those into agonists, so insulin mimics, and they have some nice properties, like they're more stable than insulin, so they last longer in the body, and they interact with a different region of the insulin receptor surface, so they're resistant to disease-causing mutations in the insulin receptor, which block interaction with insulin. So they have some interesting therapeutic properties. So the first part of my talk, I'm going to describe, or the second part, I guess, I'm going to describe applications of these methods to medicine. And so this is the TNF receptor, which is the TNF pathway is really central to inflammatory disease. And so here you see a diffusion trajectory carried out in the presence of the receptor. And the protein that gets formed is kind of an unusual protein. It's very extended, and it sort of hugs the receptor. And this protein is actually quite remarkable. It binds the receptor with single-digit picomolar affinity, and it cross-reacts with the receptors from humans, monkeys, and mice. So it makes it very easy to do experiments in model systems. This is an experiment in mice where this is a mouse model of inflammation, and this designed protein is very effective at suppressing inflammation. So we've made versions of it that are stable in the gut. They're resistant to gut proteases, so we're excited about using this protein to treat inflammatory disease. And then we're making conditional versions that can suppress inflammation at different places in the body. We are designing proteins that can activate the immune system in the opposite direction, sort of amplify the immune response for attacking cancer. And so this is a design that, like natural cytokines, brings together two cytokine receptors. But unlike cytokines, it's very, very stable. So again, it gives a prolonged immune response. And so we have collaborators at the Dana-Farber Cancer Institute who are finding very promising results with these sort of designs in treating pancreatic cancer, which is important because pancreatic cancer is a very difficult disease to treat. And one of the things, so we kind of design these proteins in my lab, we test them, but when we find things like this that look really like they could be potential therapeutics, we start companies that then can push them out in the world and do all the clinical development. So recently, we've been focusing on another aspect of the immune system, and that is the peptides, which are displayed on MHC molecules. So as you know, or as many of you know, cells have on their surfaces these MHC molecules which present peptides, which are sampled from the various the many proteins that are present inside the cell. And it's really the only way of targeting cells based on what their internal contents. And so for cancer therapy, for example, this is how neoantigens are presented. So we've been designing proteins that can read out the identity of peptides in the context of the MHC. And this is what T cell receptors do. And that's how T cell receptors recognize, T cells recognize targets. But T cell receptors are quite unstable. It's been very difficult to develop therapeutics based on TCRs, whereas these designed proteins are very, very stable. And we've been able to make designed proteins that are very selective for the cancer target, but not for closely related off targets. And for those of you who are familiar with the history of T cell therapies, this is a celebrated case where there was a therapy which targeted the MAGE cancer antigen, but there was also cross-reaction with Titin, a host protein, which was catastrophic. And we can now make designs that really only interact with MAGE and not with Titin. And importantly, they're recognizing the peptide in the context of the MHC molecule. So another important class of drug targets are GPCRs, G protein coupled receptors, which mediate many important physiological processes. And many of them are hard to drug. They have narrow recessed pockets, but we can now use these diffusion methods to design proteins which are both antagonists and agonists of GPCRs. So we're excited about these as sort of a new type of therapy for targeting the many diseases where GPCRs are implicated. Now, we can condition the process further by specifying not only what the target we want to hit to bind to is, but we can also specify the type of protein we want to generate. And so the pharmaceutical industry is particularly interested in antibodies. There's a lot of experience with antibodies. And the proteins I've showed you so far are not antibody molecules, but we can condition the design process, the generation process, and tell it to generate antibody-like structures. And so in this case, we are telling it to make VHH molecules or nanobodies, and this is the influenza surface protein, the hemagglutinin, and the generative AI generates generates proteins which bind the influenza virus, and this is a cryo-EM structure comparing the structure of the generated protein, the designed protein, to the actual protein. There seems to be like, I don't know if this would activate Windows settings. I don't know if there's a way to make that go away, but maybe it doesn't matter. So we've also used this generative AI method to design full-length antibodies. And so here's an example of the structure of a full-length antibody. Here, this is just an scFv, but we can also make it fuse this to Fc to make a full-length antibody. And here you see the structures of the individual CDR loops are very, very close to the design model. So my prediction is in 2 or 3 years, antibody immunization for making antibodies will sort of be replaced by these type of generative AI methods. Because they have the big advantage, you can target a particular region on an antigen, and you can build in other properties, for example, the developability. Oh yeah, sorry. That's a little bit of antibody. So antibodies have what are called complementary defining regions. There are 6 loops that antibodies have that are what interact with their target, and this is just showing that. So these are the regions that are different from antibody to antibody, and this is just showing that the structures of when we solve the structures of these designed antibodies and we look at each of the loops, the structures are very close to to the design model. Yeah, thanks. So there's another important class of proteins in biology which are intrinsically disordered. So everything I showed you so far was designing binders to ordered proteins, but we can also target intrinsically disordered proteins. But then the problem is, well, what structure do you target? And the answer is that we don't specify the structure in advance. So here, this black peptide is the intrinsically disordered part, and we simply have it fold up during the diffusion process. It's getting noised along with the protein that we are. So we start off with random noise, for both the protein design and for the peptide or protein we're targeting, except we keep the sequence fixed for the protein we're targeting because that's the thing we're trying to bind to. And then you can see they sort of fold up together. And so this works really well. We've been able to make binders to many different proteins and targets. This is the peptide dynorphin, which is implicated in chronic pain. We can make quite potent binders to this. Amylin, which is involved in, which forms amyloid fibrils related to diabetes, and various cancer surface markers, including, actually I talked to a student earlier today, we also are making binders for protein transcription factors like MYC and HBX that can sort of shut down the aberrant transcription they produce that causes cancer. These methods are, again, we've made these methods available. It's quite easy now to make binders to disordered proteins. So the case I wanted to tell you a little bit more is for a particularly important disordered protein, tau. So tau is a major player in neurodegeneration. It's been very closely linked to Alzheimer's disease, for example. And so we've used the methods I just described to make binders to different regions of the tau protein. It's a big protein, but they're large parts which are intrinsically disordered. So here's a picture of one of these binders, and here's the tau peptide in blue here. And so we can use these binders in different ways. I just wanted to mention one result. We can take the binder and fuse it to what's called a degron, something that induces degradation of the protein and whatever it's bound to. So if we take cells that express tau, this is just a Western blot showing you the tau, and we co-express, and this tau, when you express it in cells, in mammalian cells, will form fibrils and aggregate. And so if we express in the same cells this tau binding protein with this degron on it, it very effectively removes the tau. And so we're very excited about this as a potential way forward for neurodegenerative diseases involving tau. And our collaborators are currently testing this in animal models of tau related diseases. So I should say that there's specific forms of tau, probably some of you are aware, that are phosphorylated, that are prickly implicated in disease. And we've recently extended these methods so we can target phosphopeptides or peptides or proteins with different covalent modifications, which allows us now to specifically target the disease-causing form. Okay, so what I've talked about so far is the design of proteins that bind to that have single states. So while there was that disordered protein, that was the target. The binders we were designing were all well-folded proteins. But there are times when you don't really want your protein just to have one state. Like for an enzyme, as I'll talk about later, you need to have something that will stabilize the transition state, but also bind the substrate and then release the target. For a switch, like a light switch, or a sensor, you would like a protein that has multiple states. So we've been developing methods for designing proteins with more than one state. And here's the basic idea. So we have a protein here that is just an abstract cartoon for now that has this straight state. When there's an effector at it, it causes the protein to bend like this. And we can build nanostructures out of this protein, like this triangle here. And when the effector is added, it causes, like I said, the protein to bend. And then we get this new structure here. And so this is just a few examples where here we have a triangle protein. Here's the EM showing you the triangle. We add the effector, and now we get the square protein, just as designed here. This is another example where we can start off with a square assembly, and we add the effector, and it goes into a pentamer. So this is you might say, well, who needs to turn triangles into squares or squares into pentagons? That's not really so useful. I will give you an example on my next slide of using this for biological control, but we're very interested in using this idea for biological computing, because you can imagine now a 3D lattice of these kind of switches where they have, each protein has multiple states, and we can put information in at one end and have the system have the adjacent switches interact with each other, basically like a 3D crystal of transistors. And we think that could have very interesting potential for as a new computing device. But and I'll show you also later using this concept for sensing. But here's how we can use this for medicine. So I showed you before the protein that induced signaling by bringing two receptors together, and you might want to turn that off if you had too much signaling, like it was too much immune activation. So here you can see we've designed a switch. So when you want to turn the signal off, you add this yellow protein, which folds upon binding, drives an allosteric change, causing dissociation of two receptors. And this works really well. We can actually follow on the surface of cells the proximity of the two receptors. This is the IL-2 receptor for those of you who are experts. When we add the first protein without the yellow one, it brings them together. And then when we add the effector protein, they very rapidly come apart. So the blue and the red, the two receptors are labeled, and they rapidly dissociate. And so this causes an immediate cessation of signaling. So we can have this medicine activates the immune system, but we can turn it off very, very quickly when we add this effector protein. So we can use a similar concept to turn medicines on only when they get to the right place in the body by having them turned on. In that case, this yellow protein would be something that was only in the environment, say, of a tumor. Okay, so I've talked about applications in technology. Now I'm going to switch in medicine. Now I'm going to switch to applications in technology, and so a first application is sensors. So here are what I told you about up till now are soluble proteins, but we can also design transmembrane proteins like these beta barrels here, and we can make their interior larger and larger. And as we do that, and then when we add them to membranes, they insert into membranes. As we make them bigger and bigger, the conductance, the amount of current going through, goes higher and higher, as you'd expect. Now, we can design these. We're designing these from scratch, so we can make the pore really of any shape we want and any internal diameter. So we have a lot of control over the conductance. So now, coming back to the switching idea, we can fuse to these proteins a switch which binds a small molecule. Without the small molecule, the switch is in the open state. In the presence of the small molecule, it goes into the closed state, and when it's in the closed state, that blocks conductance through the channel. And so here, we're looking at the current going through. That's what's being measured here. And then when we add the small molecule, that completely blocks the current going through. So we're very excited about this as making sort of enabling the design of molecular noses. You could imagine designing many, many proteins like this that respond to different subsets of small molecules, kind of like our olfactory receptors do in our noses, and using this to work out what's in your environment, what types of molecules. So these proteins I've told you about are inserting into lipid membranes, but since we're designing them, we can actually make the proteins much longer and have them instead insert into polymer membranes, which gives some very nice properties because polymer membranes are much more robust than lipid membranes. We can also go even further by making holes in silicon nitride chips and designing proteins which insert into these chips. And so when you drill a hole, or there are various methods to make holes in silicon nitride, but they don't produce really, really regular holes. So it's been hard to make really consistent nanopores with them. But the design protein that fits into the hole then is very homogeneous. And so we see very stable currents after these proteins insert. And so we're interested in these both for sensing and then for applications like single molecule DNA sequencing and protein sequencing, where having an array in silicon has a lot of advantages, more direct coupling electronically, and we can pack them at much higher density than you can in a lipid membrane. So now I want to talk about applications in sustainability. And as I mentioned, one of the things we're really working on a lot now is designing new catalysts. And here we start not with a target we want to bind, but instead a ideal active site that we hypothesize would catalyze a chemical reaction that we want to catalyze. And so so we start with this. We use this all-atom version of RF diffusion to build up a protein around it. Then we make the proteins and we see whether they catalyze the chemical reactions. We've been working both on bond-making reactions, trying to, for example, come up with new ways to make pharmaceuticals. Like one of the things we're working on a lot now is making cheaper ways to make tuberculosis drugs, which need to be very low cost because they're for the developing world. And this also has the advantage that this sort of synthetic method can avoid using harsh solvents that are required for many reactions. And then Siren got us inspired to think about plastic degradation. And I should say that we can start thinking about reactions that biology really never thought about, which are like simple reactions from organic chemistry, like SN2 reactions, which those of you who took an organic chemistry course know about, where we have an electrophile and nucleophile. The protein in that case positions them in exactly the right way to combine, and we've made catalysts in that way that are very effective for a number of different non-biological reactions that aren't really catalyzed in biology. But I wanted to tell you about today in more detail is we've recently succeeded in designing proteins which break the peptide bond. Now, this is important because, well, first of all, it's a hard problem because those of you who've worked with proteins know that they're stable for a very long time in solution. The bonds that hold amino acids together don't break spontaneously. So the way we've done this is to take active sites. This is an active site that involves a metal ion, a zinc ion, and here it's being bound by 2 histine residues and a glutamate residue, and we have a couple other residues that help with moving the protons around, like taking the proton off this water molecule to activate it for attacking the peptide bond. And then we use this RF diffusion, all-atom version, to build a protein which not only holds these residues in place, but also has a binding pocket for the peptide. And so we make those proteins in the lab. In this case, we made 96 of them, and several of them had quite high activity. And I'll just show you what that looks like. Here's an example of one of those designs. This is work by Anqi Chen and Keija Wu. I guess I think their names are coming later. This enzyme requires zinc. So if we don't have zinc, nothing happens. But if we add zinc, this is a substrate molecule of protein that we've designed. This is the no design protease. This is just an SDS gel. We get stoichiometric cleavage, and it cuts at exactly the right site. And then we can mutate each of the residues that was, so this is the protein that's getting cut in the presence of zinc. And as we mutate away these catalytic site residues, we completely lose catalysis. So we can measure the specificity. We can design proteins that proteases that cut different peptides, and they're quite specific for the intended substrate. And they're very potent enzymes. They carry out many turnovers, and then the rate accelerations, like I said, are pretty pronounced. So like how good an enzyme is, how good a catalyst is like how much does it speed the chemical reaction over the rate it would have gone on without the catalyst. And so there are estimates of how long, what the spontaneous hydrolysis rate of the peptide bond is, and these designs are accelerating this by 8 orders of magnitude, which is really impressive. And it's very promising for now thinking about doing new types of chemistries with designed proteins, because we now know we can make designs that really have these profound rate accelerations. And we've solved structures of some of these proteases, and they're very similar to the design models. Here's the active site, and here's the bound zinc. And so I think the problem that we've kind of solved here is the problem of if you have an arbitrary collection of atoms that you want to position in space, we can now design proteins that will hold those atoms in position. So just a few more examples before I finish. One of the things that Hanala and I are both very excited about or fascinated by is the problem of mineralization. So Hanala will talk about teeth involves a mineralization of calcium phosphate to ultimately form enamel. And they're also in our bodies. We have bone, and then there's shells, all kinds of sea creatures that. And in all those cases, we have proteins that interact with inorganic minerals to deposit, to mediate the template, the formation of these different materials. And it's really quite fascinating. There's not only medical applications, which Hanala will describe, but there's also materials applications. Because what if we could make totally new materials that were proteins templating, for example, a semiconductor formation, or other, the proteins that are designed don't have to use earth-abundant inorganics that biology use, like calcium carbonate and calcium phosphate. We can use much more exotic materials. And we have another example that we've posted a preprint doing this for zinc oxide. But today I just wanted to show you, because it's going to tie into Hanala's talk, using these design proteins to mediate the mineralization of hydroxyapatite, calcium phosphate. So here's the basic idea. We start with a hydroxyapatite calcium phosphate lattice, and we design a protein that's chemically complementary. So here, this protein has carboxylate residues that are chelating the calciums in a layer, a facet of calcium phosphate. And we do this throughout the surface of the protein. And then here we've built this. This is work by Tracy Yu. Tracy's built a trimer here that has these carboxylates perfectly positioned to interact with the calciums in a calcium phosphate layer. So now, if she takes this protein and looks at it by negative stain, she sees the expected sort of hexagonal structure. But if she now incubates that with calcium phosphate precursors, you can see the interior is filling in. And similarly, she can design this structure to stack on itself to form this nanotube structure. Here's a cryo-EM structure of the nanotube, and you see this forms this long tube with this nice empty hole in the middle. This is in the absence of calcium phosphate. But if she incubates it with calcium phosphate, that hole fills in. And so we sort of have a new type of protein mineral composite material. In this case, it's using calcium phosphate. And I should say that when she looks at higher resolution, she sees crystalline mineral with exactly the spacing you expect for hydroxyapatite. So we're very excited now about making a whole range of new materials in this way. And again, Hanala will describe using designed proteins for teeth. And then finally, is my final example. We're very interested in photosynthesis. Is really fascinating, right? You're harvesting solar energy and using it to make and break to make new chemical bonds. That's fundamentally how all synthesis, or the energy for synthesizing new molecules, comes from. So in photosynthesis, we have two chlorophyll molecules known as the central pair, which are at the core of the photosystem. We've designed small proteins which position two chlorophyll molecules in exactly the same position, same relative geometry as they are in the special pair. So this protein now absorbs light just like the photosystem does, and we are and so we're doing several things now with this. First of all, we're designing light harvesting systems that can funnel more excited, more energy into the system. So we have large ring-like compounds. We're also designing larger assemblies of these proteins. This is a cubic assembly where each one of these blue things is this chlorophyll binding protein, and with many chlorophylls bound. And then I talked about just a moment ago about the designed enzymes. We are coupling, or what we're trying to do now, is to couple this sort of light absorbing system. So it's going to create an excited electron, or it does when light absorbs. We now would like to use that excited electron to carry out, to reduce one substrate, and then to oxidize a second substrate because we have a hole that's left behind that will pull off an electron. So in biology, in photosynthesis, it's a very complicated process with many steps where we split water on this side, and ultimately the high energy electrons go on to NADP to form NADPH. But we're really excited about using this to make light-powered reactions. So that's what I wanted to tell you about today. So I've shown you that we can now have methods for designing proteins. These methods are now, we've made everything I've described is now available, so you can get these programs and use them to design proteins yourself. I've described some applications in medicine. I talked about neurodegeneration, cancer, and immunotherapy. There's a lot of exciting applications in pandemic preparedness, for example, inhibitors and proteases for targeting pathogenic viruses. In technology, I talked about the sensors, the idea of an artificial nose, these new devices we're working towards new devices for sequencing proteins. And in sustainability, work with Siren on breaking down polymers. I talked about artificial photosynthesis. But this is just a very small set of the possible applications for protein design. And then I want to acknowledge the wonderful people that I have the great privilege of working with. And I want to thank you for your attention. And again, to everyone, to Siren and to you for inviting us to speak. Yeah.
The mic.
Thank you, Professor Baker, for sharing your insights with us. May I invite you to please take a seat on stage?
Okay.
May we also now invite Associate Professor Siren Lim from the School of Chemistry, Chemical Engineering, and Biotechnology to be the moderator for the Q&A session.
All right, good afternoon, everybody. Thank you so much again. I've never seen a lecture hall this full before. and great talk, David, as usual.
Thank you.
And now we see that so many things can be done by design proteins. And I'm pretty sure all of you are very excited about what it can do, or they can do. So I'm going to open the floor for burning questions. I have a bunch of questions. Wow, okay. Before I even start, okay. Maybe I'll take from the two young people over there, and then we'll move up to the front. Okay. Yes.
Yeah, I really enjoyed your talk. This may be a bit of a weird question, but can this denoising be mapped to the evolution of life to see how amino acids became proteins in the end?
Oh, that's a very interesting question. Well, there's nothing. So the method is being trained on the structures in the protein structure data bank, right? So it's basically a way of so the process doesn't the denoising process is simply the way the neural network has figured out how to generate more things that look like what's in the protein structure data bank. So I don't think there's really a direct parallel to evolution, because to understand evolution and to model evolution, I think it's a really interesting idea, and I think you could use deep learning methods. You can use deep learning methods to try and simulate the evolution of protein sequences over time, and it could be a similar sort of denoising process. But in the way we're doing it, it's really just a way of generating new protein structures sort of outside of evolution, particularly since the proteins we're generating are really very different from, are not proteins that came through evolution. So I think you could use deep learning to do what you're describing, to simulate protein evolution, but that's different from what we're doing here.
Thank you.
One more question up there,
Yeah.
woman.
Thank you so much for the talk. My question is, when you design a new protein and until the final goal of it proving therapeutic efficacy, I assume that there might be a loop, right? The AI design and then wet lab synthesis, and you would have that. How does that loop run? And are there steps for you to improve the speed of that, wet lab, dry lab iteration?
Yeah, that's a really good question. And so it's different in different cases, but particularly for enzymes where you would like to have very high activity, like Siren and I have talked about, degrading polyproline or something really, really hard. You would almost certainly, we don't really know how to design an enzyme to do that now. So if we did try, we probably wouldn't get very high activity to start with. And so we've been working in parallel on methods for making the iteration, just like you described, you were right on, between experiment and computation more faster and faster. So that means faster ways to go from making DNA DNA fragments that encode your design proteins to actual proteins. And I think Siren's setting that up in her lab now. And then also ways of very rapidly assembling different possible versions of designs, or many different designs, into many different proteins, assaying their activities, and then doing active learning to sort of iteratively optimize. So you're absolutely right. There's a lot to this that is not just what's going on the computer. There's a lot of experimental back end that I didn't describe. And as we get to harder and harder problems, I think we're going to need to do more and more of that. And so I think more of the focus, the AI part, will be going into active learning. How do you iterate? There's sort of the engineering part. How do you rapidly iterate experimentally between or integrate experiment with computation, which I think is a lot of the work we're doing now on enzyme design, is sort of trying to figure out those problems. So it's a very good question.
I just wanted to mention one thing, that this is something about BioFoundry, which we actually set up also in Singapore, a National Center for Engineering Biology, which actually trying to do this in big scale in terms of synthesis. And 2 questions from the front, please, here.
I have a small question and a big question. The small question is when you use AI to design a molecule or protein, you can very carefully design how does it fit into the receptor, but then the other side of the molecule may produce some effect that you may not expect to find, because these are all synthetic and doesn't come from natural evolution. Would there be some complication or side effect of the drugs that you may not actually expect to happen?
Well, that could be. So immunogenicity is the main thing that you would worry about, that you're putting a foreign protein into the body, and there would be a worry that it would cause an immune response. And so we haven't seen large immune responses. So some of these design proteins have been in humans. Many more of them have been in animals. And so far, we haven't seen any responses that are where it looks like they're toxic or things. But I think the answer, I mean, in reality, you would always check, right? So you would, if you were trying to develop a new medicine, the original, how you get that protein is different from traditional, say, antibody discovery. But once you have it, there's the same sort of toxicity testing, safety testing, efficacy testing that you would have for, say, an antibody drug. So I think, so the answer is, yes, it's absolutely possible that something else could be going on the back end. We haven't seen evidence of that, but that doesn't mean it couldn't happen.
The big question is, do you see all the big pharmas will in the future all use AI to develop biologics instead of using the traditional biological methods?
Yeah, I think that within 3 years, the world will have switched over to using these AI methods to largely to make antibodies, just because you can target specific regions. I mean, maybe it won't be 3 years, maybe it'll be 5 years, but it's cheaper. You don't need, and it's it's much more controlled, and you don't need to don't need to inject things into animals. You don't need as much material of your antigen. So I think we'll see a wholesale shift. It hasn't happened yet. So pharma companies are looking at this kind of technology, but they're not. But it's not the way it's done today. But I think it will be, without exception, the way it's done within 5 years. And it's not just my group. So my group's obviously an academic group, but now there are companies that are developing methods too. So there's a lot of activity in this space.
Yeah, David, very nice talk. So just on a related question, what about the ethics? Where do we draw the line? And what kind of questions that we should, as academics, that we should sort of set the direction for these pharma companies?
Yeah, well, wait, what was the last part of your question?
What kind of guidelines or maybe rules that we should define as academics for this pharma company?
I think, like with any new technology, there's always worries that something dangerous could be done, for example. My own personal view, and so when we develop new software, we consult with colleagues in the community before we release it. My own personal view is that there are a lot of dangerous things. Like, think about the 1918 Spanish flu. It just devastated the world. The decision was made to publish that sequence, as many of you know, some number of years ago. So anyone can go and make the 1918 Spanish flu. Now, that's the DNA sequence. You still have to make it, make the virus, but you could put it into a mammalian cell and you'd have produced it, which is really scary. But there was sort of a communal decision to go ahead with it. Well, there's nothing that anyone can design with protein design today that approaches the complexity or dangerousness of a pandemic virus, because I showed you examples of most of what I showed you was binding and catalysis, but something like a virus has to bind to a cell, it has to get into the cell, it has to get its genetic and material replicated, it has to make many copies of itself, and then it has to get out. So there's a lot of very dangerous stuff that's already in the public domain. So my feeling right now is that these methods are much better suited. There's much more due to prevent nasty things like pandemic viruses. Humans don't really have to make new ones because nature already perfected that. There are also many toxins like botulinum toxin and so forth. So I think it's an important question to keep in mind. I think there are other ethical questions in biology that are probably more pressing, like gene editing babies. Hanel and I were talking about this earlier on, or embryos, So, but it's an important question. Safety is an important question to keep in mind.
More question? One here.
Yeah, thank you for the inspiring talk. So I'm interested in quantum properties of crystalline materials.
Yeah.
So I want to create like new superconductors, magnetic semiconductors, topological materials. What's your perspective on using these generative AI methods to create new crystals? Is there something special about working with proteins, that you have some kind of restricted, chemical space that makes these methods very successful? Or who do you think is doing the most interesting work?
I guess there's two-part answer to that. One is if there was a particular crystal form of, the methods I described here for calcium phosphate could really be applied to any binary, anything that you thought could form a particular crystal, and you could template the formation of particular polymorphs. So that would be using this mineralization approach. As far as materials design, I had a very interesting conversation about that yesterday. There have been, reports of AI methods for developing crystals. I think it's still a little bit controversial whether new materials how effectively they really can be designed. But in terms of accessing new types of materials using the protein design methods, I think that's pretty clear that that's possible.
Thank you.
Okay, more questions? All right, there are so many questions. So 1, 2, 3, and 4. All right, gotta go.
Thank you for your wonderful talks. So I'm a new PhD student, which starts to focusing using AI to generate new proteins. So we can use deep learning method to generate many protein and peptides. But like when I have many candidates with good APTM or low energy state, so I don't know which one should I choose to do the later validation. So I may ask you for the some opinion. And if the metrics are not good, so what's your preferred way to optimize? Whether generate a larger library and screen, or any more elegant way to optimize?
Well, yeah, I mean, those are good questions. They really are problem dependent. So how do you, if that RF diffusion sort of animations I was showing, those are quick to run on a computer. It's just a minute or two. So you can do this many, many times, and so you can generate many, many candidate designs.
Yes.
And the question is, how do you pick which ones to experimentally test? And that really depends on the problem. And in some cases, there are pretty good ways to evaluate. Like for binder designs, the simplest way to do it is to predict the structure of your designed protein with the target, and see whether it's predicted to bind just exactly as you designed. For an enzyme, particularly an enzyme that's carrying out some new chemistry, it's much harder to evaluate, and that's where it comes back to this active learning, where you're iterating between experiment and computation. As far as how do you improve, what do you do in the case where you're not getting designs that have the geometry you want? Well, fundamentally, you probably need newer methods. So that's why we're always trying to improve the methods. So RFD3, for example, is much better at complex constraint satisfaction than RF diffusion was. And like you said, you can make more designs and then screen through for the ones you want. It really depends on the case, I would say.
Great, thank you.
Okay, thanks. So there, over there, 1, 2, and then 3.
Hi, David. Excellent talk. So it's very interesting that you pointed out that there are many cases that doesn't work as well. So I can't help but ask, like, what are the common failure modes of the current AI tools that you build, and what are your thoughts on how we can improve those and close the gap?
Yeah. So we don't always know what the failure modes are. I mean, and it really depends on the problem. So, for example, in the protease case, some fraction of the time the proteins don't express. You make the protein in bacteria, but it doesn't actually get made, or the protein gets made and it simply doesn't have activity. But what's always been true is every time we start working on a new problem, the success rate is low. But then after we work on it some time, now with the proteases, and Anqi Chen, who did the protease design work, she made another round, and there over 50% of them were active. So it's sort of understanding. So we don't know. The problem is it's hard to know about failure modes, because it's the only way you could really know would be, like, it might be that the protein might not have folded at all. It might have folded up to a protein with a different structure, or it might have folded up to a protein with a structure that's exactly like what you wanted, but your hypothesis that would catalyze the chemical reaction was wrong. So it's a little bit hard to debug that, because you need to solve structures of each of the designs, and that's quite time consuming. So it's kind of like the Anna Karenina quote, like protein designs that don't work can all not work for different reasons, but the designs that work tend to work for the reason that you designed.
Okay, great. So then one question there, and then one more there, and then anybody else on this side of the room. Okay. I think we might need to, okay.
Cool, thank you so much for the interesting talk. My question is around predicting protein stability, because of course for it to carry out the intended function, it has to be stable in that environmental conditions, which may be not stable, but might be changing. So do you have any way within your machine learning models to essentially help you predict a protein that will be stable in the desired environment, I guess is my question.
Well, that's an interesting question. It turns out empirically that the proteins we design, and this was also true back in the day when we were designing proteins with Rosetta using sort of physically based models. One of the things we found that we were surprised at, many years ago when we first designed the first de novo proteins, was that they were very stable. We could often boil them and they were fine. And that's held true in the deep learning era, that the proteins that we design are almost always very, very stable. And you might think that's very surprising until you consider that in nature, there's not really been selection for stability. Proteins just have to function under the conditions where they operate. So in a human cell, for example, they have to be stable at 37 degrees, but you never see 47 degrees, higher temperatures. And proteins in nature are constantly being hit by mutation. So if a mutation brings the stability down, but not to a point where it compromises function, it doesn't matter. So I guess the first part of answer to your question is that design proteins almost always are very, very stable. If we need to make them more stable, we can do that. But sometimes we want proteins, like we're interested a lot now in pH-dependent proteins that will be folded under, that will bind to a target at high pH, but at low pH, say in the endosome, will let go, or in the lysosome will let go. And so there, we want to design instability, but only under certain conditions. And that we do by making hydrogen bond networks with histidine residues, which are pH dependent. So yes, so we can generally design proteins are stable. We can design in conditional stability where we would like it.
Okay. I got like a sign that I need to wrap up right now, and then we have like a lot of questions. We have questions from the audience who are pre-submitted, and I felt like I should probably address at least one of them. And I would, a question coming from an NTU staff from Center for IT Services and Laura Lee. And with the increasing dependency on AI, what is the most valuable trait or value that professionals can contribute to research and delivery of medicine? Would such dependency actually create a crippling of research and healthcare delivery systems, and how we think you can overcome that?
Well, let's see, why would there be a crippling?
Perhaps because you have all these assistants that relieve human from actually thinking.
Oh, well, humans still have to think. In fact, yeah. So when it's sort of interesting, this isn't an exact answer to that question, but when we switched over from using sort of the physically based methods, Rosetta, to the deep learning methods, people asked me, well, how could we switch over so quickly? And that's because everything really changed. Everything changed, but really everything stayed the same. So you set up, the problems we were trying to solve were more or less the same, and the way we thought about that was the same, but just we were changing out what was under the hood. It was instead of a physical model, it was an AI model. So the human thinking and the science all stayed the same, which is why we could develop new methods, plug them in, and and it was pretty straightforward to do. What was the first part? Or maybe that's...
The increasing dependency, what is the most valuable trait and value that professionals can contribute?
Well, I think the most valuable trait, it's like science just generally. I mean, there's science is all about discovering the unknown, and applying these methods and design proteins in all these areas is totally new. So I would say the scientific spirit, sort of discovery. In the case of working towards therapeutics, obviously you need to be very systematic and rigorous and careful. You need, you don't want to put anything into humans before it's passed all the safety trials. Fortunately, the FDA and the Singapore version of the FDA are going to look at design proteins just as stringently, probably more stringently than other proteins. So I think but the big picture answer to that question is just being great, just doing great science is the most important thing.
And then the human is still the core, I suppose.
Yeah, yes, human's still at the core.
With that, thank you very much, David. And then please give a round of applause.
Thank you, Professor Baker and Professor Lim. Next, we have our second distinguished speaker of the day, Professor Hanale Rohola Baker, Co-Director, Institute of Stem Cell and Regenerative Medicine, University of Washington. The title of her lecture is AI-Driven Foundational Regenerative Medicine. Please join me in welcoming Professor Rohola Baker on stage with a warm round of applause.
Thank you very much. It's great to see so many young people in this room. Thank you for the organizers and Syrian for inviting us here and giving me a chance of giving a few stories from my lab as well. We came here 2 days ago, and maybe this is familiar tower for you. Just when we landed on the Singapore airport, sort of simple thought came to my mind, but that thought was pretty dramatic. What if this aviation tower would only react when planes crash? No guidance, just when planes crash, that's when they would be helping the situation. That would be pretty risky, right? Probably we wouldn't fly if that would be the situation. However, today, that is exactly what modern medicine is still doing. We respond when disease has already crushed us, has caused problems in our body. In some cases, there are incurable problems at that point. So today, I'd like to talk to you about a new idea. I would like to talk to you about how this is the time to change so that modern medicine doesn't only act when human disease has happened. And this new method that we want to talk about is AI-driven foundational regenerative medicine. And really the idea is to change from reactive care to programmable regenerative medicine. It is the AI firms or AI-driven foundational regenerative medicine's goal to be all about control, being that biological control tower. So that we can act before we get the disease destroying our body. Why do we think that we can do that? I think time is right now. We all probably have realized that there's been 3 dramatic revolutions that has happened during the last 10 years. One of them is AI that now allows us, many of us, to work towards generating virtual cells. These virtual cells then can be used to predict biology and generate trajectories. That was revolution number 1. Revolution number 2 is, just like you heard, AI-designed proteins. We can now generate proteins on purpose, generate proteins that do what we want them to do to control cell fate. We can generate them to heal, to change the fate of the cell. We can generate them to regenerate. The third revolution really is stem cell and organoid biology. We can now take these fantastic proteins and introduce them to stem cells, and now make the stem cells and their progeny to be the healers, the regenerators, the changers of cell fate. And result of this all, we think, is proactive programming of the repair and regeneration. So how would that then work in a disease? Let's take an example. Actually many diseases are cell fate disorders. For example, if it's neuron, if this is a neuronal cell fate disorder, we could now take our AI firm and build a virtual neuron. Basically, we would begin from iPSCs, induced pluripotent stem cells, and generate a model of that disease, that neuronal disease, by using iPSC-derived neurons. We would analyze them in such deep level that we can now simulate and predict what the cell would do in later disease state. That we call virtual cell. Many of us are working hard to create virtual cells for different cell types. Virtual cell then would allow us to really define when help is needed. What happens before the crash has taken place? We could create a protein that can be the guider, the guiding post. And that is what the protein design would allow us to do. We can make protein with intent to avoid the crash. This would now create programmable biological instructors. And we believe it is really generating a new operating system, new way of guiding biology, not only reacting when biology has gone wrong. And we hope this in the future is really changing the clinical medicine. So bottom line is that even rare diseases perhaps become solvable. Okay, you might say, this is great, so we need to do regeneration, but don't we, as our bodies, don't we already regenerate? And we do. All you young people, your stem cells are regenerating as we sit here continuously. And that is fantastic. Unfortunately, though, when we get older, our stem cells stop regenerating. And that creates many problems. It creates multiple problems of diseases that are really aging and enriched in aged population. These are unmedical needs in aging populations. And today we'll talk about 4 of such unmet medical needs. Vascular stability. This causes dramatic strokes, and now we believe microstrokes actually are an initiator of Alzheimer's. Diabetes has lots of wound healing problems due to vascular problems. Second or unmet medical need is neuronal pain perception. Neuronal pain is a dramatic problem that causes then opioid overuse in many cases. The third problem in aging population is muscle regeneration doesn't happen. We get sarcopenia, and as many of you know, that causes lots of problems. And the fourth problem that I want to bring up here is tooth regeneration. When we get older, we don't only get caries, but we lose our teeth. So my lab has now for a long time actually tried to work on the rules of regeneration, tried to understand how stem cells can help us regenerate, how we can learn to make those old stem cells regenerate. We've worked on early pluripotency. We've shown that metabolism is very important for those cells. We've created disease models in regeneration, for example, for sudden infant death, and shown that cardiomyocytes are very important in that case. However, around 10 years ago, all of that work on regeneration really led us into a bottleneck. That bottleneck was that we can not make the cells to become to the level that we see in our bodies. You take iPSCs, these pluripotent stem cell-based cells, and you differentiate them. They invariably go to fetal stage, but to get them really become the cells, the muscles that you young people have in your adult bodies is very, very hard. We realized something has to happen. We need to have some new tools. And that's when we connected to IPD and Baker Lab and really began a dream team. The goal of this team is to design novel proteins using this AI-based model that David just talked to you about, and use them to govern regeneration either by receptor binders or epibinders. And of course, today we are very happy we did that 10 years ago, because just last year, Demis Hassabis and John Champer from Google got a Nobel Prize on structure prediction, and David Baker, our own David Baker in UW, got a Nobel Prize in protein design. And I am not shy. I can brag that party was actually pretty amazing, and I even got to sit next to the King Carl Gustav. So, but this then seriously got us very far in utilizing the designed proteins. There was a question here, how do you go about? What's the pipeline? We use the protein design pipeline for IBD. And in this, we have a pipeline, very important part of that is RF diffusion. And there are multiple steps to then create multiple designed proteins for epibinders, which in our case would be binders that bind to epigenetic regulators and therefore DNA and gene expression. And then on the other hand, our goal was also to identify receptor binders that will bind the receptors and affect signaling. Today, I'll talk to you more about the receptor binders and their effect. So protein design in regeneration is real thing today. I'd like to now go back to those 4 examples that I mentioned, those unmet medical needs in aging population. And I'm going to give you an example of each as far as using protein design to try to solve some of these problems. As I mentioned, we of course first long time worked on naturally existing activators, ligands. However, many of them are unstable, non-specific, and difficult to manufacture. And designed proteins really solved those issues. So I promised that I'll give you 4 examples. Let's begin with the first example, vascular stability. There is a well-known receptor pathway called Ty2. Ty2 is involved in vascular stability. However, it has multiple ligands, and it was not clear what the key regulators of that pathway are. Now, with help of Baker Lab, we created scaffolds and added the ligands at the end of these scaffolds. And from that kind of work, we very clearly showed that Ty2 receptor requires high valency ligands for introducing vascular stability. That wasn't quite good enough, though, because in this case, we still used the natural protein, the ligand binding site that binds to the receptor. That's no good. In terms of utilizing this for perhaps human clinical cases, because now you are introducing a natural protein and very likely you do create immunity, and in that case that would be disastrous. So we wanted to create fully designed protein, fully designed Ty2 mini binder. And with, as David said multiple times, that some of the proteins are hard. This was a very hard target. Only after RF diffusion 2 came about, a binder was generated that bind very tightly to the receptor and was a very good inhibitor. We mentioned that this is a binder. It binds to the receptor surface, and it is an inhibitor. Of course, that's not quite what you can use for for medicine. We wanted to create an activator. And now we have learned that these high valency situations can make an antagonist to an agonist. And we showed that this truly happened also in the case of Ty2. Now remember, when you make a designed protein, it can be very specific. In this case, in this particular complex and the signaling pathway, usually the ligand binds not only with one receptor, Ty2, but also binds with integrin, and other receptor. This designed protein, however, does not bind the second receptor, yet it is active. We further showed that in this case, the designed protein internalizes very fast to endosomes in the cells, suggesting that maybe we solved a long-standing biological problem here also, that integrin function probably is to stabilize, keep the signal longer going on. Might this designed protein-generated agonist then act as a real thing? Is that enough to activate the pathway in biological context? Is there biological therapeutic value for this designed agonist? We tested this in a flu model. In flu challenge, unfortunately, many of the mice die. But when Ty2 agonist was given through nasal spray, we showed some therapeutic potential. That is that fully designed Ty2 agonist can prevent the mortality of these influenza-infected mice. So we argue that it actually can combat flu-induced ARDS, acute respiratory problem. So in the future, this truly might be so in a way, two things were solved in this case. We showed that the fully designed Ty2 agonist can act on its own. It's specific. It doesn't need integrin, and it has a potential therapeutic capacity. It can act without that co-receptor integrin. So that was one vascular example. Second vascular example is FGF receptor agonist. Again, we created high valency structure, and that acted as an activator of FGF receptor. And in this case, we showed that utilizing this can create vascular cells that are particularly biased towards arterial fate, and arterial vasculature is, of course, the vasculature that comes from our heart and brings the oxygenated blood to our tissue. So that was one example of vascular stability and how designed proteins can potentially show some therapeutic value in the future. Our second example is going to be in neuronal pain. As we understand, neuronal chronic pain is a very dramatic problem and particularly can cause opioid overuse in older individuals. Well, it turns out that there are some families, there's some genetic backgrounds in which the families don't feel pain. People have studied these families for some time. One of the families has shown to have a mutation in NGF, nerve growth factor. Nerve growth factor is actually today used in clinics. It is used for neurotrophic keratitis, but it's very, very expensive. And NGF has a problem. You can use NGF, but when you do so, neurons do grow, but you have a dramatic neuronal pain. Clearly, that family that had a mutation in NGF did avoid the second part. And people wanted to understand what was the mutation in NGF in that family. They identified one specific point mutation. That point mutation binds to a receptor P75. This ligand, NGF, is just like all of our naturally existing ligands. It is not specific. It binds to two different receptors. It binds to TrkA, that here in two components of TRAC-A, or it binds to TRAC-A and P75 hetero structure. Since this family that had a mutation for P75 binding did not feel NGF-induced pain, now an arms race began to try to create a protein that would do just that. Itself, the NGF with the mutation is very badly behaved, and it really took a protein design revolution to create that protein that will now only bind to TRAC-A and not P75. Shown here, it binds TRAC-A but not P75. It also activates that TRAC-A. And big question was, would that create pain? So far, our answers are no. So this TRAC-A agonist actually reduces substance P and G and other markers for pain, and it has low excitability. So we think this truly could be now our painless NGF, and we are very excited of taking this forward for preclinical work. So that was number 2. Our example of using designed protein number 3 comes from muscle regeneration. As you know, your muscles are healthy when you are young. They lose their strength when you get older, unfortunately, due to sarcopenia. I today have talked about using designed protein as antagonists, or David has talked about them also using them as antagonists for receptor signaling. I've talked about how high valency can make them agonists. Now, with the muscle regeneration, I would like to introduce a totally new kind of modality. And in that, what we did, we took 2 of the mini binders, binders that bind different kind of receptors, and we fused them together. We call these novokines. So instead of binding, we take a binder that binds to FGF receptor, for example, and we combine it with a binder that binds to TRAC-A, and now these novokines will bring 2 receptors together that usually we think don't interact in a cell. From big screen of these kind of novokines, we found a handful of really interesting molecules. Today, I'd like to talk about one. This novokine shown here brings HER2 next to FGF receptor. This new pair of receptors is extremely useful for muscle regeneration. I show here one example where we use H2F in something called transdifferentiation assay, where fibroblasts can be induced to take muscle cell fate. None of the existing ligands work in this process. However, this designed novokine can make very massive and strong muscles. And furthermore, muscles need to have sarcomeres. And these sarcomeres are the ones that will allow your muscle movement. And with the novokine, the sarcomeres are very prominent and exciting. And we furthermore decided to use patient or patient-derived myoblasts, and we added this novokine to that situation and found that the sarcomeres in these new myofibers were dramatically better and the force was stronger. So in the future, we hope that when your parents are beginning to lose their muscle mass, maybe H2, this kind of novokine, in the future can be injected into the muscle. They activate the myofiber fusion and hopefully regenerate muscle in this case. In that case, we wanted to understand if this truly becomes a therapy, we need to understand the mechanism. And we showed that this both receptors are required for the activity. We showed that with kinase nulls that while FGF receptor kinase domain is essential, HER2F, while important, essential component of this complex, its kinase domain is not functioning in this case. We've showed that many effectors that are known to act downstream of FGF receptor did act here as well, except the calcium signaling. While FGF induces calcium signaling, this H2F novokine did not. So based on this, we have shown that this novokine is a biased FGF receptor agonist. So with many of these experiments, we have now shown that novokines are functional. They can be, actually in this case, truly, this is demonstrated that synthetic receptors can not only pair, but rewire new kind of signaling pathways. And they might have new functions. Like in this case, the idea that this could really be a therapeutic tool for regenerating muscle is totally novel. We're very excited about this. This doesn't only stay in muscle regeneration. This kind of novokines could be a programmable platform for regenerating other tissues as well. So I've told you 3 examples. 4th example is our teeth. All of you have beautiful teeth with enamel, the white material on the top of the teeth. That is the strongest material in our body. However, all of you, or most of you, also have cavities. We lose some of the enamel. Over 90% of humans have lost some of the enamel. We get cavities, and that is the beginning of the cycle of death of your tooth, unfortunately. Why doesn't our body then regenerate that? That is because our body does not anymore have those cells that can make enamel. Our solution for this big problem is that we again use iPSC, induced pluripotent stem cell-based differentiation protocol. We have set up a protocol so that we can make ameloblasts, and that was great. However, again, we were stuck in fetal states. We only could go to the mature state where secretion was seen by using designed proteins. Example is shown here. This is a soluble notch agonist, and by using this particular designed protein, we now can make cells, organoids, that can secrete calcified material that has the qualities of enamel. This enamel is secreted in apical side of these cells, and that's exactly what we need. However, we are not quite done yet. So we can make enamel, it's calcified, but it is not as strong yet as your enamel. And our hope, our goal really is to use designed protein, this particular designed protein that David talked to you about, a hydroxyapatite inducer protein, to create the enamel fibers exactly so that we can create enamel that has the strength of your existing enamel in the teeth. And really the goal, sort of big goal here, is to regenerate, of course, and eventually to make new teeth. But let's begin by regenerating enamel. We believe that's a new frontier in living dentistry. And this really allows us to create, has allowed us to create a breakthrough platform. Utilizing designed proteins, we now can produce these cells that are mature enough to secrete enamel to the apical side of these cells. This enamel needs more help by these hydroxyapatite inducers, but we are very close. Clearly, there's massive unmet need. In this case, enamel loss in all of our case is irreversible, and this problem is global. Globally, people have problems with their teeth. And today, what happens? We drill, when there's a small cavity, we drill more, and we add batches of different materials into our teeth. That is not living. Our goal is to really create living fillings. Enamel producing ameloblast layers so that we can regenerate our teeth instead of initiating the tooth cycle of death. So those were the 4 examples I wanted to give you today. I hope that with these examples I have convinced you that we are living a new era of biology. We don't only stop in watching the crashes happen, the diseases happening in our body that are become then irreversible. We hope that now in the future with these designed proteins, we can really be guiding the process. This is a guiding biology in which hopefully your aging is much healthier than your parents' aging. And I've given 4 examples of such, and of course we hope we can create many more examples. And we definitely feel that the time for regenerative medicine with the designed protein and AI is today, and hopefully you all join that. And I'd like to thank many people in the lab and our collaborators for all of this work, and I thank you for your attention.
So prepare to disconnect the laptop.
Thank you.
Hit over to the chair.
Thank you, Professor Rohala Baker, for sharing your insights with us. May I invite you to please take a seat on stage? May I now also invite Associate Professor Sia Kalin from the School of Physical and Mathematical Sciences to be the moderator for the Q&A.
This is a really fantastic talk. I think all audience here will agree that behind any successful gentleman, there is a highly motivated and inspiring lady. Yeah. Yeah. Okay, I think we can directly jump into the process. Any questions, please raise your hand, and then we'll pass on. Yeah. Yeah, with a T-shirt, yeah.
First of all, thanks for the talk. It was very fascinating for both of the session. So in your cases, in the enamel growing case, how do you identify which protein to target first? Because when you want to design the protein to bind to a certain target, you have to first have the target that you know that, if we design something that binds to this target, it would lead to enamel regenerations. But because the enamel regeneration is a pretty complicated process, right? So how do you design it first, and how do you improve upon it? Because you already have a design in mind already, what is the next protein you want to target, right? Yeah, that's my question.
Excellent question. Excellent question. Absolutely, the whole process is very complex. But remember, though protein design is fantastic, we are not only relying in protein design. We are relying in 3 revolutions: AI revolution, a protein design revolution, and stem cell revolution. So there are multiple. So first we took the iPSCs, and those we can differentiate to all different cell types in our body. That's why we can now make first time ameloblasts. And so ameloblasts themselves, they secrete multiple compounds that are, so actually you should say, we can make ameloblasts that are fetal, but then we needed designed proteins to really push them forward. And we did know particular pathways that were active at that stage, but they just didn't get activated by normal natural ligands. And in that case, we knew, for example, the soluble NOTS agonist really helped push to the maturation state. But even that isn't quite enough. So in this case now, the cells are mature. They're secreting many compounds, or many proteins that are needed for enamel biogenesis. But at the end, again, this designed hydroxyapatite inducer is totally essential to create the structure that would give strength to the enamel. So biology shows the problem, and we acted upon that.
And have you explored other types of perturbation as well, like small molecule perturbation or like gene knockout perturbation? Does those also kind of can be helped?
Right. So absolutely, you can use CRISPR. You can use small molecule to repress some signaling pathways. None of them are, none of these small molecules that we've used have been as specific as the designed proteins. So the idea is really to remember that Singapore airport, Kanchi, the aviation tower. Really, the idea is to use as good aviation, as good guidance as possible, and you're absolutely right. Many other ways. We have many other ways at the moment to regulate cells. Just none of them are as don't give us as good precision as designed proteins do today.
Thank you.
Fascinating talk. In case of the HER2 and FGR growth factor receptor, yes. Is it a known fact that when you put the two receptor together, it promotes sarcomere?
Yeah, that's a really good point. So designed proteins, you can say they are useful for regeneration, but designed proteins also create something totally new, can answer new, can create something that we didn't expect existing. So in this case, no, it was not known that HER2 and FGF receptor as a pair can be useful for muscle regeneration. We didn't even know that those two could ever signal together. So in this particular case, designed proteins actually answered something very new in biology as well. I think this is a whole new avenue that we can take now. We can create these novokines and test them not only in regeneration, but others have already begun or tested them in immune cells and so on. So that this is new and very exciting.
A follow-on question, how did you target this two receptor? How did you discover that this would promote sarcomere growth? And if you didn't know ahead of time, what was your process to try to find this new pathway?
Excellent question. So the idea of putting two mini-binders together and testing whether you get the signaling activity from that was just the idea. You had to have a screening method to see if anything happens. So in our case, we use this transdifferentiation fibroblast to muscle cell transdifferentiation as an 8-day screening method. We screened hundreds of novokines, and only few of them were very powerful in this case. So biological screen is what we use to really tell us whether these novokines can do anything if they can induce regeneration in biology, then they pass the question of that it's worth of analyzing them on mechanistic level. Yeah,
Thank you.
thanks.
Thank you. I think the lady on the back. Yeah.
Professor, thanks for your showing the exciting samples, the application in the medicine. So I would like to know how close are we to deciding the protein design to the clinical application? Yeah, thank you.
So your question is, how close are we actually getting some of these into clinic, right?
Yeah.
So that's a good question, and we push that very hard. David's lab clearly pushes that very hard. We push that very hard. There are many of them are in preclinical tests right now, and I probably would be lying if I give you a year when they can be in clinics, but our hope is that we get some of these going to clinics in a few years of time.
Okay, thank you. A following question. What kind of the disease will be the most possibility to become the first clinical application for the AI driving protein design the medicine? Which disease will be the first or the most possibility one to using the protein design medicine?
Right. That's also a very good question. That what is the lowest hanging fruit, basically, right? And it is possible that that something like tooth enamel regeneration might become the first one because it is quite safe. Many others have to go through many of the safety. Of course, that has to go through safety as well. But you will not then inject the designed protein into your body. You now give the cells and the enamel to the teeth. And hopefully also, and certainly David has talked about cancer. There's a big push for getting these designed proteins in cancer trials, and those are quite far. In regenerative medicine, I think things like if these novokines truly help in the animal experiments as well, these preclinical studies, it will be very exciting to see if we can use those to regenerate our aging muscles when the normal regeneration doesn't take place anymore.
Thanks for your.
Thank you.
We have a. Okay, maybe you. Yeah.
Hi, Professor. Thank you for the wonderful talk. I'm currently a student, neuroscience student, working with stem cells for neurodegeneration. So I'm very curious. What do you see as the major distinction between using stem cell organoids, the actual ones, and the AI-assisted virtual cells, for example? And what do you see in the future, how they complement each other?
Yeah, very good question. So my guess is that we certainly have to make organoids because we don't have virtual examples of organoids yet. So the process of creating virtual cell is still ongoing in many places. Now to be able to create virtual organoid is yet another level of complexity. So really lots of work in getting to virtual cell level, not only one kind of virtual cell, but multiple kinds of virtual cells. But after that, our hope really is that then the simulation can happen in virtual life, simulation of a disease. We want to identify a stage when the first things go wrong. the situation when the crash hasn't happened yet, situation when guidance is needed so that the landing can be successful. And that's the place where I think, again, the protein design becomes a very important guider. So if simulation suggests that at a particular stage, now particular signaling is abnormal, now we can make a designed protein on purpose that will affect at that early stage. And hopefully that would be a way how we guide also in individuals where we know that mutation exists, try to guide the process so that we can avoid the problem forming in the tissue. Sure.
Yes, I think we can have follow-up questions that for PhD students who are interested in regenerative medicines, what skills they need to have now. For instance, like the AI people now need to learn more knowledge about stem cell, and then for people start stem cell, they need to learn more knowledge about AI. So what do you recommendations for the skill set for these students interested in that?
Do everything. Absolutely. This is the time of not just staying in your comfort zone. This is the time to try to reach and see what connections you can make because we are beyond just singular processes. We need to understand and govern many things.
I think there's another question is follow-up is that, during the tea session, you give a very beautiful discussion about, how you driven into study the science, right? We have a lot of, young students here, and the question is that what are the, inspirations, what are motivate you goes into science? And, can you give us some guidance, what are the right track we should, put in mind? Yeah.
Right. So my history is I'm coming from a small village from Finland. That village, it's a farming village. Nobody knew about science there. There were educators who made me believe that one can do more. And I went to big city, Helsinki. It's not that big, but it is big for a farmer girl. And in that city, I was in university and asked the professor a question that the professor said, that's a good question. Why don't you come to my lab and test this? And I did an experiment and I was totally hooked. I realized that science is just the highest form of humanity. It feels like you do art every day. You create, you get to play with your ideas. You get to produce something that didn't exist. You are the first one to see the result of your experiment, either in virtual experiment or real experiment. And I hope we all value that, what we do. It's a cream of the cream. It's a very, very special way of living your life. So enjoy every day in the science.
Thank you. Yeah, maybe I'll use my chance as the moderator. So, actually, I have a political question about that discussion with David yesterday. Is that now AI people are super optimistic, right? We will solve all the medicine problems, all the cancers within a decade. Biological people on the other side is a little bit pessimistic. Okay, biology is so complicated. Everything is, and in your last slide, you mentioned about the time is now, right? So I think you are more towards the optimist side, and can you give us some comment on that? Yeah.
I'm optimistic. I have seen some science. I've seen stem cell work for many, many years, and I think just now we are in the inflection point. We really can do something today that we couldn't do 5 years ago, and I think that's going to make a big difference.
Totally agree. Yeah. Thank you very much. Yeah. Let's think. Yeah.
Thank you, professors and distinguished guests. This brings us to the end of the IAS Lee Gong Chen Distinguished Professor Public Lecture. I would like to thank you all for your kind presence. May I request all to remain seated while our speakers, guests, and senior management take their leave first. Lastly, we request attendees to fill in the feedback form by scanning the QR code on the screen. Your comments will help us improve our future events. Thank you once again for taking the time to join us today, and there are refreshments served at the open area outside the auditorium. Have a lovely evening ahead.